15.3, Synaptic Communication. Just a moment ago in the previous segment we initiated an action potential. An action potential occurs when a neuron reaches threshold depolarization that opens voltage-gated sodium channels, and we have movement of ions across the membrane, and this creates a wave of activity that's going to continue the entire length of the axon, and it terminates at the end of the axon, which is the axon bulb. The axon bulb can communicate with other neurons or glands or muscles, and this point of communication is called the synapse. So in this diagram, you see there in red the presynaptic neuron. So that was the neuron in that previous segment that we stimulated the threshold. So we have this action potential depolarization wave rushing at 250 miles an hour until it reaches the axon bulb. So that neuron that's sending the message is the presynaptic neuron. The cell that is receiving the message would be the postsynaptic neuron, or this postsynaptic cell could also be a muscle cell or a gland. Now notice in the diagram these cells don't quite touch one another. There is a little space which is called the synaptic cleft, and there is a chemical messenger that's going to be released by the presynaptic cell that has to diffuse across that synaptic cleft. So we want to look a little bit more closely at that synaptic cleft, and these chemicals are neurotransmitters that are going to be released across the synaptic cleft. So this is an enlargement of that diagram. Here, kind of on the middle left, you see the presynaptic neuron, the axon bulb. This is the one that's sending the message. Whenever the action potential reaches the axon bulb of that presynaptic neuron, it causes some voltage-gated calcium channels to open. Those voltage-gated calcium channels allow calcium to come into the cell, and that allows some secretory vesicles to fuse with the plasma membrane. The secretory vesicles contain neurotransmitters. They're packaged in little membrane vesicles, and if they merge with the plasma membrane, those neurotransmitters are going to be released into the synaptic cleft. As they diffuse across the synaptic cleft, they will come in contact with the membrane of the postsynaptic cell, which is over on the right side of the diagram now. And this is just really cool. On the membrane of those postsynaptic neurons, there are some receptors that bind the neurotransmitter. And would it surprise you to learn that those receptors are also sodium channels? Whenever the neurotransmitter binds to that receptor, they open and they function as sodium channels. And what was it that started this whole process in the last segment? We allowed sodium into the cell. That changed the resting potential across the membrane. If it reached threshold, it initiated an action potential. So if we have enough neurotransmitter on this postsynaptic membrane and we open enough of these neurotransmitter-gated sodium channels, we can initiate an action potential in the postsynaptic neuron as well. And so neurons function in these multi-cell circuits where one neuron has an action potential, it communicates with another across the synapse by releasing neurotransmitter, and then we can initiate the action potential in that next neuron in the circuit. There's a lot going on at these points of synaptic communication, however. If you look at the top right of this diagram, this is a picture through a scanning electron microscope. You see the cell body of a neuron, and all of those structures on the outside are axon terminals or axon bulbs. Look at all of them. So you have all of these different neurons trying to communicate with this guy. So this is really like the little cartoons you see where somebody has the devil sitting on one shoulder and a little angel sitting on the other. And so the devil's saying, go, 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 do it. And the little angel's like, no, you know better. And so really the same type of thing is happening here, because you can have either an excitatory or an inhibitory stimulus or message delivered to this postsynaptic cell. Some of these that are communicating with this cell may be releasing neurotransmitters that open sodium channels. That would be excitatory. If we allow sodium to come into that postsynaptic cell, that's going to push it closer to threshold, closer to initiating its own action potential. But some of these neurotransmitters are inhibitory. What if instead of opening a sodium channel, we open a chloride channel? Chloride is negative, and if we allow negative chloride to move into that postsynaptic cell, that's going to make it more negative on the inside. That would push it further from threshold, and that would inhibit that cell from initiating a nerve impulse. So the activity of this postsynaptic cell has to take into consideration all of this input from sometimes many, many different axons from different presynaptic neurons.